So this is going to be chapter 8, looking at patterns of inheritance. So Mendel, who is considered the father of genetics, was the person who uncovered a lot of the fundamentals of genetics. And what he experimented were with garden pea plants. So this is just a picture of Mendel, and he was a monk. So in his process of studying genetics, what he examined was the colors of the flowers on the pea plants was initially what he studied. He looked at plants that had violet flowers and some that had white flowers. And he crossed two true breeding plants. A true breeding plant is one that's consistently going to always produce the same as the parental variety when it is self-pollinated or cross-pollinated with the same type of flowers. So a true breeding purple or violet would always produce violet flowers. A true breeding white plant would always produce the white flowers. So when he did that, the parent generation is called your pea generation. He crossed a true breeding violet flower with a true breeding white flower. And it's really important to note that what he got when in the F1 generation, which is your first filial generation, it means son, is all progeny that had violet flowers. This discredited the common thinking at the time was that traits blended together like paint colors. If that would have been the case, they all would have been light violet instead of violet flowers. It helped to prove that traits were actually inherited as discrete units. So then when he did self-fertilization of this F1 generation plant, what he got was about a 3-to-1 ratio of violet flowers to white flowers. So we're going to look at Punnett squares and how that happens in this unit. So when he studied the pea plants, there were 7 characteristics that he used in studying them. He looked at their seed shape, were they round or wrinkled? The seed color, were they yellow or green? The flower color, purple or white? The pod shape, inflated or constricted? The pod color, yellow or green? The stem height, tall or dwarf? And the flower position, axial or terminal. So when we're looking at genetics, we look at the phenotype and the genotype. The phenotype are the physical traits that are expressed. It's what you actually see in the organism. It's the traits that are transmitted by the alleles. The alleles are the variations that are possible, such as the flower color. The genotype is going to be the genetic makeup, the ratio in the gene combinations of the offspring. Sometimes the genotype is going to be the same as what you see in the phenotype. Other types, the genotype will be a mix and you're only going to see one of them. When you're looking at dominant and recessive alleles, capital letters are going to represent the dominant alleles and small letters will represent the recessive alleles. So here at the top you've got a parent generation where you're crossing a yellow and a green seed. The yellow is going to be the recessive trait, so we represent it with 2 small letters. The green is the dominant trait. We're representing it with 2 big letters. Both of the letters are the same because these are going to be true breeding plants. So when you do the cross, what you're going to see is all of them that are going to be yellow. And actually on here, what you have is the yellow is the dominant trait, and they're just backwards on here. So that's why you have yellow. So 100% are yellow. And the reason you have that is what you're going to take is you're going to have one of the alleles is going to come from this parent. One of the alleles will come from the other parent. So you're going to, each of them will get one big Y, one small y. Being that yellow is dominant, it's going to be the trait that's expressed in 100% of those flowers. So when you do the self-fertilization again of this F1 generation, what you're going to see is about a 3-to-1 ratio. 3 of them will be yellow for every one that's green. So 25% are going to inherit 2 big Ys, 50% will get one big Y, one small y, and 25% are going to get two small y's. Two big Y's and one big Y, one small y are both going to have the appearance of being yellow. The only way to get the recessive allele expressed is to get both of the recessive genes. So the allele for albinism is expressed in humans, but it's recessive. So what that means is both of the parents have to carry the recessive allele, and both of them have to get passed on to the offspring. So albinism is seen in all races of humans. So genes are going to be transmitted on the chromosomes. And the reason you're going to inherit one from one parent and one from the other is based on the way that we divide the chromosomes in meiosis. Meiosis is similar to mitosis, but it's going to have a second dividing phase. So you start out with interphase. This is the cell's normal interphase that we studied before with cell division. Here you're going to go through prophase, metaphase, anaphase, and telophase 1. So during prophase, you're going to actually form the chromosomes. You're going to start to dissolve the nuclear membrane. Pro-metaphase 1, they're starting to get lined up, and in metaphase 1, they're lined up along the metaphase plate. In anaphase 1, they're going to separate, and in telophase, you're going to have two daughter cells. So when you do a second division of meiosis, you will have each of these daughter cells split. And instead of copying the genetic information, they're going to split that genetic information in half. So here we can see a test cross that's going to be performed when you have an organism expressing a dominant trait. A homozygote is going to be one that has two of the same traits. A heterozygote will have two different traits. So in this test cross here, we have a yellow seed, and we know with yellow that it can have a big Y and a small y, or that it can have two big Ys. We're going to cross it with a green plant, or a green seed here, and we know that has two small Ys. So when we do this test cross here, if we're going to assume that this one is a big Y, what you're going to get for all four of them is a yellow plant. The way that you fill this in is you take the gametes, the two possible gametes each parent can make. So the yellow parent makes a big Y, and here we're doing the test with it being another big Y. The green parent has two small y's, so you have two small y's. You're going to bring each of these down into the box. So what you see here is the small y goes here, the big Y goes here. Small y goes here, big Y goes here. Over here, you're going to bring the big Y down, the small y down. Same thing here. So this test cross, if you had all of the offspring being yellow, it would say that the parent is homozygous dominant, meaning this parent was homozygous two big Ys, which are the dominant trait. The other possibility is that this parent has a big Y and a small y, even though they're still yellow. So when you bring this down on these, both of these, you're going to get a big Y and a small y. They will be yellow. Over here, when you bring both of the gametes down, they'll both be small Ys. And again here, both will be small Ys. So if this parent is heterozygous, they're going to have a 1-to-1 ratio of 50% yellow offspring, 50% green offspring. So we call this a Punnett Square that's going to show this cross between the yellow seeds and the green seeds. I did put in some additional video links for you to watch that shows how to do these Punnett Squares, as well as the explanations that are here in the notes. It's not unusual for people to want to see a couple different explanations of them. So here you're going to have a cross between the true breeding parental generation of plants, two big Ys, two small Ys. The only gamete this one can make is going to have a big Y. The only gamete this one can make is going to be a small y. You can't make what you don't have. So when these are going to form their offspring for the F1 generation, what you're going to get is one big Y, one small y, and it will be yellow. So then when you self-pollinate this generation here, what you're going to get is a gamete that's one big Y, one small y. So when you create the Punnett square for these, here you're bringing two big Y's down into the square, one big Y, one small y, one big Y, one small y. All three of those will be yellow. Here you've got two green that are going to come down. So this is where you get the 3-to-1 ratio in the F2 generation. As long as you know how to do a Punnett square and make these, it's not so important that you remember necessarily having this 3-to-1 ratio. If you know how to do a Punnett square, you can always just write it out, and it doesn't take long to figure it out. Because if you have parents that are not true breeding, you will get different ratios. So this can be done looking at more than one trait, and we call this a dihybrid cross when you do more than one trait. They do have to be on separate chromosomes for this to work. So here you've got one that's yellow and round. Round is dominant. Here you've got one that's green and wrinkled. Wrinkled is recessive. So when you cross these P generations here, what you're going to get is the F1 generation that will have one big Y, one small y, one big R, one small r. And they're going to be yellow and round. So when you look at all the possible gametes that come from this, the easy way to figure it out is if you think of doing the FOIL method for this. So you're going to take the first of each letter, a big Y and a big R that come from right here and here. You're going to take the outer two, a big Y and a small r that you've got right here. The inner two letters, a small y and a big R, you've got right here, and then the last of each letter. So first, outer, inner, and last is where you get the FOIL mnemonic from. So that small y and the small r end up right here. You do the same thing for the second parent because it's going to provide one that's the sperm, one that's the egg. So then you bring all four of these letters down into the box, and you'll fill in each of those. So what you'll find in here is you're going to get a ratio of 9 that are yellow and round, 3 that are green and round, 3 that are yellow and wrinkled, 1 that is green and wrinkled. So you have the 9 to 3 to 3 to 1 F2 phenotypic ratio. You are going to have some practice problems to do with these. I strongly recommend doing at least all of those that I give you. If you are going through this and you feel like you want extra practice, don't hesitate to let me know and I can send you more. So the reason this happens is because you're going to have each of the alleles independently segregate when you form the daughter nuclei. So here in metaphase 1, you can see we've got a blue chromosome and a red chromosome. When these chromosomes are going to separate, you've got a 50-50 shot of the blue one ending up here, a 50-50 shot of the red ending up here. When the second one separates, a 50-50 shot, blue is going to go this way, 50-50 shot, red will go this way. So then when these are going to separate, you're going to get 50-50 shot, one of these goes here, one will go here. 50-50 shot again, one goes here, one goes here. On here you end up with the blue ones being separated. So metaphase 1, metaphase 2. The other option is that you get blue goes one way here, on this one you get red. Over here one you get red, one you get blue. So what you're going to end up with is a mix of gametes here. So you're going to have a different possible combination. So for each chromosome, it is random 50-50 chance which of the two you're going to pass on to the gametes. So occasionally we do see things have incomplete dominance. These are pink flowers of a heterozygote snapdragon that result from incomplete dominance. So one parent was red, one was white, and the offspring, because of incomplete dominance, is going to be a mix of the two. When we look at codominance, this is what we use for blood types. With blood types, we use the letter I, and we use a superscript for the letter for the gene. So somebody who is type A is going to inherit a type A marker. So here you have a type A from one parent and a type A from the other parent that will come together to make type A. This parent here that's up on the top is type AB. This one is type AB as well. So over here, this gamete is going to pass on the B. This one passes on the A. What you get is an A and B. This is going to be a type AB individual. This will be a type A individual. Here you get an AB individual, and down here in the corner you get a type B individual. So you're going to actually express both of the markers that are on the cells for blood types. So we call it codominance. So when we look at all the possible blood types in here, you can have the IA that's going to be type A. You can have one IA and one small I that's a type A. You can have a small I and an IA that's also type A. Type O is where the small I comes into place. It does not have any marker on it. So if there's no marker, it's type O. If it's type A, it's gotten the A marker from one or both of the parents. If it's type B, it's gotten the B marker from either one or both of the parents. When it's type O, it didn't get an A marker or a B marker from either parent. When it's type AB, it got a marker A from one parent, a marker B from the other parent. So there are things that are also linked on the sex chromosomes. When it's on the X and Y chromosome, the appearance and distribution will be different between the males and the females. What was studied for this is the Drosophila, which is a fruit fly. They only have 4 genes, they breed prolifically, they were easy to study. So the way they looked at this was having the wild type and the mutant type of phenotypes. Red eye color was the wild type, and it was dominant to the white eye color. So when we looked at the fruit fly here you have the male, and on the X chromosome is your trait. There is not one on the Y chromosome. Here you have the female. She has the W on both of her X chromosomes for white eyes. So you have your female gametes. Her 2 possible gametes that she can produce are right here, XW and XW. For the male, you have XW and Y. So when you bring them down together, 2 X's, that's going to be a female. Both of them are going, one will be a big W, one will be a small W. Same thing over here. One will be a big W, one will be a small W. So because the red is dominant, you're going to see red eyes. For the male, you're going to have an X and a Y chromosome. Because this one gets the XW and no other second one to be dominant over it, whatever it inherits on the X is going to be what it expresses. So this one will have white eyes, and this one will also have white eyes. So in addition to moving the entire gene, we can actually have more variation occur through crossing over. So what happens is you've got your homologous chromosomes aligned. We've got genes A, B, and C, A, B, and C. Here's your homologous chromosome with A, B, and C, and A, B, and C. When you have crossing over, what's going to happen is part of the homologous chromosome is going to switch with the neighbor. and it will make a new recombinant chromosome here that is part of the original part of the one it swapped with. So here you've got two different chromosomes from the parents that will actually get paired on and put into the gametes. This allows for a much greater amount of genetic variation. They figure in a human each chromosome is going to have between 1 and 3 crossing over events where this will happen. Another thing we can have happen with genes is called epistasis. This is going to be when one gene is going to affect the expression of another one. So this is showing the example here with mice. This is also how Labrador dogs get their colors. You have black labs, yellow labs, and chocolate labs. So in the seal, you have the C allele here that is going to say we're going to express the coat color. And then you have the A allele here for coat color. So A is going to determine, is it carrying a black coat, a brown coat, or is it an albino? C is going to say whether or not you're going to put that coat color into the mouse. So in that sense, if you have a gene to deposit coat color, it will be determined on do you have big A's or small A's. If your gene says you're not going to deposit coat color, you're going to be white and not deposit the coat color at all.